Hello everyone, this is Group 3. Our group members are Kwong, Justin, and Ting Wei. Today I'm going to talk about the genetic algorithm and show you guys how it works. I think after this presentation, you guys will get some idea about the genetic algorithm and know how to use it to solve some problem. Let me show you the content of this presentation. Definition, principle, background, application, conclusion. I'm going to do the definition and introduce some principle and give you guys some terminology about the genetic algorithm. My other teammate will introduce the background and give you guys some good applications in the real world and give you the conclusion. First question, what is a genetic algorithm? Genetic algorithm is a search-based optimization technique based on the principle of the genetic and the natural selection. They are used to generate some high-quality solution for optimization problem and search problems. It is a subset of a larger branch of the computation known as evolutionary computation. It was developed by John Holland and his team. So let's see the next one, the genetic features. In genetic algorithm, we have a collection or population of the possible solution. And each of the individual, each of the chromosome is assigned a fitness value, and the fitter individual are given a higher chance to mate and keep more fitter individuals. And for example, like this, we are going to initialization the population, and then we evaluate the fitness value. And then we do the recombination and mutation, like a natural genetics, producing a new children. And the process will repeat over various generations, which means the selection, crossover, mutation will repeat over and over again. For example, like this, we pick up some genes from parent 1 and pick up some genes from parent 2 and do the crossover and produce offspring. Why do we use GA? I think the main reason we use GA is we can get a global maximum solution and not a local maximum solution. We can get closer to the best result. This makes genetic algorithm attractive for use in solving the optimization problem. I give one example. We can use a genetic algorithm to solve some difficult problem. In computer science, there are a large set of the problem which called NP-hard. And even though you use some most powerful computation system, it will take a longer time to solve one problem. So if you use a GA, it can provide a near optimal solution in a short time. In other reason, the failure of the gradient-based method. You know, the gradient-based method is a traditional calculus-based method. So they work by starting at a random point and moving in the direction of the gradient until we find the maximum or minimum point at the hill. We can see this graph. We randomly pick up the point and we want to find the maximum solution. So we just go up. And then we found the peak of the hill, right? Found the maximum value. They think this is a global optimum solution, but it's not true, right? So it means it works for single-picked objective function, like a cost function in linear regression. And if it is really complex problem, there are so many peaks and valleys, which cause method to fail. It can get a local maximum solution, can only get a local maximum solution. So if you use a GA, you can get a global maximum solution. let me introduce some basic terminologies. Population, it is a subset of the possible solution to the given problem. And we can see this graph, we can see the picture. There are so many individuals, right, in the collection of the population. Each of the individual is a chromosome. Chromosome is one such solution to the given problem. And each of the chromosome consists of the genes. And each of the genes, there is a value in each of the genes we call the allele. Phenotype and genotype. Phenotype is a population in the actual real world space in which the solutions are represented in a way they are represented in a real world situation. And genotype is a population in the computation space. And the solutions are represented in a way which can be easily to understand and manipulate using the computing system. Decoding and encoding. When we get some solution in the phenotype, we want the computer system to do some computing. We need to encoding those solutions. And after the computing, we can use the decoding, convert from the genospace tab to the phenotype. Fitness function, a function which takes a solutions as input and produce the suitability of the solution as output. It means each of the chromosome, we put each of the chromosome into the fitness function, and then we get a fitness value for each of the chromosome. Genetic operator, there are so many genetic operator, for example, like mutation, selection, and crossover. Those alter the composition of the offspring to produce a new generation. For example, like this is a crossover example. Pick up some genes from parent 1 and 2 and do the crossover to produce a new offspring. Let's see this basic structure. You know, all the genetic algorithm will follow this basic structure. And the first step is population initialization. Population size should not be very large, as it can slow down the GA, cause a slowdown. And there are two primary methods to initialize the population. One is random initialization and a heuristic initialization. And the random initialization populate the initial population with completely random solution. And the other one is populate the initial population use a known heuristic for the problem. But there is a problem, right? When you use a heuristic initialization for all the chromosomes of the population, it will cause one issues is each of the chromosomes will be really similar. It may cause the premature convergence. This is not good for it. It will impact the final optimal solution. It's not good because in the genetic algorithm, we have to keep the diversity of the population. The best practice is first, at the beginning, we use a heuristic initialization and to initialize some population and then fill it up with the random initialization. Combine those two types together. Fitness function. Each of the solution need to be awarded a score, which called fitness value, indicate how close it comes to meeting the overall specification of the desired solution. And in the fitness function, there are two kinds of function. And the first one is fitness function, and the other one is objective function. If in a small or simple problem, the fitness function and the objective function are the same. If in a complex problem, the fitness function will combine with multiple objective function use together, combine those together. There are two key things. You have to make sure the fitness function will be efficiency. It will be really fast to compute. If the function is really slow, it will slow down the GA. And in addition, it must be quantitatively measure how fit a given solution is. If you get a solution, if you get a fitness value, it cannot describe how fit the solution is. You know this fitness function is not correct. It's not good. Parent selection is the process of selecting parents which mate and recombine to create offspring for the next generation. Usually, the parent solution is used based on the fitness value produced from the fitness function calculation. The key point is parent selection should maintain the good diversity and to prevent one extremely fit solution from taking over entire population in a few generations. It will cause a premature convergence. It's not good. The last one is make sure the amount of the chromosomes which are selected are equal to the original amount of the population. What does that mean? At the beginning, for example, at the beginning, you set up 10 chromosomes in the population. After the parent selection, you also have to select the 10 chromosomes. The amount should be the same. Violate well selection is one type of the parent selection. And how to use it? There is a table. Each of the chromosomes in here and their fitness value, right? We have to sum the fitness value first, and then use each of the fitness value divided by the sum of the fitness value, which you can get the proportion rate for each of the chromosomes. And then you set up the fixed point and spin the roulette wheel. When the percentage when each of the chromosomes in front of the fixed point, you can select it. We usually use the cumulative probability. What's that mean? Calculate the sum of the fitness value, first way, right? And first step, and then randomly generate a number between 0 and S. This is a fixed point. The second step is fixed point. And starting from the top of the population, adding those fitness into the partial sum P. Add those until the P exceeds the S, and that chromosome will be selected. And other two types, one is SUS. It's really similar to the previous one, but we have to set up the two fixed point, which means you can select the two parents chromosome at the same time. And the other one is a tournament selection. You randomly pick up several chromosomes and compare all those chromosomes based on the fitness value and pick up the best as a parent. Crossover operator is similar to the reproduction and biological crossover. In genetic algorithm, each of the solutions generally represented as a string of the binary numbers, which called the chromosome. So there are several types of chromosome: one-point crossover, multi-point crossover, and uniform crossover. This is third step. One-point crossover. In this one-point crossover, a random crossover point is selected, and the tail of the both parents will swap and produce two new offspring, right? And the multi-point randomly pick up the 2 point and swap some segment of them and produce a new offspring. The last one is a uniform crossover. We don't divide the chromosome into the segment. We just treat each of the gene separately and determine each of the gene, each pair of the gene will be pick up or kick out. Mutation can be defined as a small random tweak or update in the chromosome to get a new solution. For example, like this type, beta flip mutation. Randomly pick up a gene and flip it to 0. You get a new offspring. Swap mutation. Randomly pick up the 2 gene and swap it, get a new offspring. Scramble mutation. Pick up the substring of the string of the binary code and shuffle it, put it back. You get a new one. Inversion mutation. Pick up the substring of the string of the binary code and inverse that and put it back. You get a new offspring. The purpose of the mutation: to introduce the diversity into the sampled population and preventing the population of the chromosome to become too similar to each other. And it will cause the premature convergence. It's not good for the final optimum solution. The GA-Silverware selection. This selection policy determines which individual are to be kicked out, which are to be kept in the next generation. It's a next, and the key point is the fittest individual will not be kicked out of the population, will be kept all the time. And diversity should be maintained because diversity will impact the final optimal solution. So we have to care about the diversity. Let's see this picture. This is a really great example. We can see all the chromosome in here and all the fitness value in here. We can see the P1, P9, and P10. The fitness value are smaller than others, and we generate a new offspring, which has have a good fitness value, right? 8 and 9. So we definitely will use those new offspring, replace the old one. We will replace a P10, pick up one between the P1 and P9 randomly, because P1 and P9 have a same fitness value and generate the new population. Termination condition. The termination condition of the genetic algorithm is important to determine when a genetic algorithm will end. And usually we keep one of the following determination. The first one is if there is no improvement in population, which means you cannot get a better solution, it will terminate, it will end. When we reach an absolute number of the solution, when you writing a code, at the beginning, the designer may set up a maximum generation, maybe 50. When you reach the 50 generation, it will automatically end. The last one is objective function. Value has reached a certain predefined value. It's similar to the second one. Let's see the pseudocode of the genetic algorithm. Actually, this pseudocode is really basic and clear. Each of the steps correspond to each of the phases of the structure. Initialize the population corresponds to initialization population. Evaluate each candidate, which corresponds to the fitness function calculation, get the fitness value, right? Parent selection. When we get a fitness value, we should perform the parent selection. And then we do the crossover mutation produce some new offspring. And then for the new offspring, we have to do the fitness function calculation again to generate the fitness value for the new offspring. And use a new, we need to compare those new offspring to the old parents based on the fitness value to determine which one will be kicked out, which one will be kept, and then do the survivor selection. When we hit the terminate condition, and we will end the while loop and return the best value.
Hi guys, my name is Kuhn. Today me and Justin are going to present the applications part for the presentation. There are many different topics that genetic algorithm can apply. In this presentation, we will focus only two topics. The first one is natural sciences, mathematics, and computer science. I will discuss about image manipulation. There are several ways to do image manipulation. For example, image enhancement, which is increase the quality of the image segmentation, grayscaling or lower the image quality, and reproducing image, which is reproducing image from a certain original image. Second topic that we will cover in this presentation is biological sciences and bioinformatics. For this one, I will cover the RNA and RNA interaction prediction application, whereas Justin will focus on DNA analysis application. Now let's go to the reproducing image. Images may consist of two-dimensional arrays, where each elements are pixels of the images. The pixel has typically 3 or 4 components. The most common one is the RGB one, which stands for red, green, and blue. We can apply mathematical calculation on those pixels to change the pixel value, which lead to the change of the image. Genetic algorithm provide the systematic random search is able to search for vital needed pixel in order to change the image in the way that we want. Reproducing image application follow the basic step of genetic algorithm. First of all, you have the original image as an input. Those data will be initialized as population. Then we apply finish function to that one. Select the passport after we finish the finish function. Then apply the crossover and mutation function. This process gonna be repeated until we have the picture that is as close as the original picture, or we reach a certain iterations of conditions that we set above. In the end, we will get the result of our manipulated image. Apply genetic algorithm into this one. So initialize population. First of all, you need to get the shape of the image to two-dimensional array or maybe one-dimensional array. Then you set up an image, set up a matrix with random pixel as a start. After that, you apply fitness function. For this fitness function, you need to find the mean of absolute differences between the random pixel matrix and the target matrix. Then we will subtract that from the summation of all elements in the target matrix. We only want to choose the quality that's higher than a target, then store it into a matrix called a quality matrix for later processing. We only choose the maximum value out of the quality matrix in each iteration. Remember that we need to cancel it out in the next iteration so that we don't recalculate it again. For the crossover function, select a certain number of genes in the target matrix and random matrix. Then we put together in the offspring. In this example, as you can see, the green one is the target matrix and the random one is the yellow one is the random matrix. We will get half of the target matrix and half of the random matrix and put together to get our offsprings. The parents from previous states are kept in case the offspring only inherits bad gene from the parents or the later offsprings. Good genes get replaced by the worst genes. This means that when the parents, the green and the yellow parents, will always be kept for at least one iteration, so that if the offspring has only the bad genes, it will get reverted. Mutation. Apply improvement calculation to the matrix. Avoid changing the percentage of mutation too high because it will create new genes that are more different and comparing to the previous one. As the result, you can see this is our original image. At the first iteration, we will create a random matrix that is basically nothing beside a very simple pixel. At the 10,000 iteration, you can see the lion image is starting to form. At a 50,000 iteration, you can see the image of the lions is you can see the image of the lions better. At 100,000 iterations, the comparing to the 50,000 iterations, this one looks better. So if we keep going, and there will be a time that the lion is kind of same with the original image, but it took a lot of time. So for our presentation, we just stop at 100,000. So talk about the time complexity of this one. In each iteration, it takes all I times j, where I is the width of the image and j is the height of the image. And for this application, the total time is going to be of k times I times j, where k is the number of iterations that we set in the conditions. Some use cases of image reproducing. We can use this image reproducing to censor an image to hide our identity or information. Moreover, we can use this as a way to store and check password, which is Face ID or fingerprint. You guys can remember that whenever you use a Face ID, whenever you set up a Face ID, it took several steps to fully cover your face. So each step is using genetic algorithm to generate a certain type of your face, and then whenever you open your iPhone, it just need to go through of the all of the generated image and look for the correct one, and then you can able to open your iPhone. Next one, I want to go into interaction prediction between RNA and RNA. RNA is a single-stranded chain of nucleotide units like adenine A, cytosine C, guanine G, and uracil U. A will connect with U by 2 hydrogen bonding, whereas G will connect with C by 3 hydrogen bonding. RNA secondary structure is presented by a list of nucleotide bases paired by hydrogen bonding within its nucleotide sequence. When we stack these pairs together, it will form a scaffolding trimming of RNA three-dimensional secondary structures. So what is the secondary structures? Secondary structures of interacting RNAs, or we can call it as SSIR, is essential for modeling RNA structures and understanding their functional mechanism. We have some examples on some structures over here. So you can see as duplex, it is a RNA connect to an RNA, or hairpin is one RNA and it connects to itself where there's a loop in the end. For 90 degrees, and we have others interaction like 90 degrees kissing loops, open three-way junction, four-way junction, or we can stack them up together with three-way stack junction or three-way loops. As you can see on the first two lines, it's only two of the RNAs or the RNAs itself connect to each other. But on the last four, there's gonna be more than two RNAs gonna be connected to each other. And we want to this genetic algorithm will try to predict which one is the best model. The interaction prediction between RNA and RNA follow the basic steps of genetic algorithm, just like the image reproducing one. But for this one, we will ask RNA sequence, two RNA sequences as our input. Then it will apply the process of genetic algorithm, which is initialize population, fitness function, select the best pool, then apply crossover and mutation function. The best possible RNA has been created and cannot be changed anymore, or the loops reach the number of iteration that set will be the conditions to repeat this RNA and RNA application. Our result will be the best folding one and the interactions of the two RNAs. So the best folding one and interactions, basically the structures that you guys have seen before on the previous page. So go to the initialized population. First of all, we start with two RNA sequences, R prime and R double prime. The length of them are N and M. So we can convert it into R star, which is combine R prime and double prime together. We will have R star with the length of K, where it is M plus N, and we need to plus 1 in the end in order to do the concat by itself. We can see over here each R star is going to be R prime of I, where I is from 1 to N, or it's going to be R double prime I minus 1 minus N, and for I in this situation is from N plus 2 to L. After that, we can create a dot matrix where the axes are R and reverse R. Over here, we can see that the pairs AU and UA is basically what I mentioned above. CG and GC also. You can see there's an extra one, UG and GU. This is a special one, and we usually these bonding are not going to be formed, so we might eliminate it during the process. So as you can see, the Ri and R1 minus J plus 1 are the ith and L minus J plus 1 nucleotides in the sequence. So we can construct the R to show all of the subdiagonals in the dot matrix. So with this form, we can exact stems and hybrid regions of the dot matrix to obtain secondary structure of interacting RNAs, SSIR. For each parent, a d to the r tuple is randomly made as I is equals to x from 1 to x dr, and x will be either 0 or 1. After that, we have parents constructed. So parents will be ck, where xk equals 1 or ck is not empty. Based on those subset of subdiagonal, we can extract stems and hybrid regions of the dot matrix to obtain SSIR, which is secondary structure of interacting RNAs. So let's go to the finish function. Pick out two random structure and then call them M and N. Find the sum of the minimum free energy to get the finish function, which is basically MFE is the minimum free energy of M plus MFE, minimum free energy of N, to get our finish function. Then we will start those pairs up after calculating fitness function and pick the lowest minimum free energy, because the lower the value, the better the structure. After that, we will need to apply the crossover function. Basically, select two parents, then concat them together to create a long RNA sequence. After that, we will apply mutation into it. Suppose that there's a parent P is already get concatenated in the crossover. We can construct a DR, which is kind of same as the initialized population. After that, we randomly place xi equals 0 or xi equals 1 to 0. So, we will replace xi equals 0 to 1 and xi equals 1 to 0. Keep doing these steps when we get a defined number of generations. So go to the time complexity of this one. It is going to be O of L squared plus the length of P. So in here, L is the total length of the two RNAs combined, and P is the length of the individual parents on the next generations. Some use cases of this RNA and RNA interaction is that you can use this one to find out how a disease is formed in order to find a way to solve it. We can also optimize treatment for certain disease like cancer or gout. And that's the end of the applications part from me.
So the second application we're going to talk about is called DNA implementation. This is applying genetic algorithms to actual DNA processes that could be conducted in labs. So you might ask, why DNA? So first, it's inspired by the pointwise mutation and crossover functions of genetic algorithms. As we discussed, those are the main components of forming an algorithm in this form. Another concern is that the computing time for DNA is proportional to the number of generations required. So this means that when you apply genetic algorithms to actual DNA lab results, the time it takes to process it could actually be done a lot quicker than if we did it through a computer process. Another similar thing is that DNA computing processes in parallel can be used for larger populations, and this parallel way of doing things allows it to be done quicker, as discussed. In addition, we can apply this to DNA because of the information storage available. For example, 1 gram of DNA can equal 2 by 10 to the 21st bits. So the example problem that we're going to talk about that is used in DNA genetic algorithms is the max ones problem. So it starts with the binary bit string, so it's just basically a string of zeros and ones, and then an initial population of these. The target and the solution is to evolve these strings to match a specified target. So it's another bit string, and it's usually just like all ones. So I'm first going to give a general overview of the whole process of how it solves this problem, and then we will be more specific with certain problems. So the step 1 will be problem initialization, and we start with just random DNA sequences as we see here. And then we predefine a target strand. So the target strand on the bottom will be the goal we need to find. step 2, we evaluate the fitness by hybridizing to target strands. So as you can see here, the target strand is on the top, and hybridization will lead to the target strand being combined with the several candidates. So there's one on the bottom with more of like the curves defined, and then one of the straight. So basically it's connecting those with the target strand, and we use the same target strand for both. So doing this, we develop potential candidates to solve our problem. So step 3, we're going to select and purify more fit candidates. So the purification is the process of removing the target strand from the candidates as we did in the previous step. And then these candidates will be used to breed. I'll also go into more detail about the purification process after the overview. So after that, we go to step 4, which is we amplify fit candidates with pointwise mutation and reserve a portion. So we're going to reserve a portion in the end, as we will keep separate sets for breeding and to have a clean set without breeding applied. So we go to step 5 now, which is when we do the breeding between candidates, and we're going to use a process called crossover. And as we said before, we see that we keep a reserve set, which is 50%, and then the breeded set on the right. So after step 5, we go to step 6, which is combining reserved and bred candidates, obtaining a new generation. So basically this is grouping up the candidates that were not bred with the ones that were, and then we just combine them together and we create this set, which is the new generation, and this is what we use for the next iteration of the whole process. So that's the overall view of the DNA implementation, and I will now talk about the specifics part of them. First of all, we will do the candidate solutions and the target. So this is what an example of what it would look like. As you can see on the top, you see target. So that'll be the strand that starts with the 5" to the 3". And then on the bottom, we have the candidate. You can notice that it's a little bit longer than the target candidate because part of the process is splitting the candidates apart, so we need to make it longer to account for that. You'll notice that there are things called clamps on the top and the bottom of these DNA strands, and basically that's to provide a fixed set, so when we're breeding it is we have similar sizes for each kind. So one of the important processes that is used in the fitness evaluation is called denaturing gradient gel electrophoresis. It will be DGGE to make it short. And for this, the target strands are that's what the process is for hybridizing the target strands with the candidate strands. One of the important things about this is that the process and time that it takes to do this is determined by the amount of denaturation in the genes. To explain, as you can see on the right of the diagram, at the top is the most denatured versus towards the bottom it's not as denatured. If the more it's denatured, that means the more time it's going to take to put the strands together. Obviously, because it's more, basically it's like a wire. If it's more tangled, it'll take longer to put together. So as discussed, I talked about this thing called DNA purification. So it's kind of done similarly to what we explained before, and it's done through conventional denaturing gel electrophoresis. So this is basically kind of the opposite of the step before. Instead of combining the target strand with the candidates, we are actually now removing the target strand from the candidates. So once we do that, we reconnect the removed candidates together. From this, we separate them into a reserve and breeding set. So the reserve sets are not going to be altered and used for breeding, and then the breeding sets are obviously going to be used for breeding. So the breeding process is done using single point crossover. So breeding strands are double stranded, like similar to a DNA, but are cut to only one strand at random locations. So this means, say you have just a sequence of 2 strands. At random locations, we're gonna just make it 1 strand. We're gonna cut out pieces of the double strand and get 1 from each. So we're gonna do this and cut the strands and combine them with similar amounts from the reserve set. So this is why we have the reserve sets, because we need to have that extra amount of DNA to combine with the ones that we are going to use for breeding. And then the final product is called the single point crossover. And then as a result, since we got both cut strands from the reserved and the breeding set, we have information from both parents, and it will be used in the next iteration. It's basically our new initial set for the problem. And repeating this process, we eventually get a candidate or a set that is similar to the target. So that is basically one type of problem that we can solve using a DNA implementation of genetic algorithms. There are also 2 different problems that could be solved. They're kind of similar but have little bit differences from the Max ones. So the first one will be the Royal Road problem. So one of the interesting aspects of these type of problems is that they allow theoretical predictions. So this basically means that, as described, we can set up a problem and see if we can find a solution, because the solution is predefined. This means that when we describe the target strand, we already predefined a solution, which makes it a theoretical prediction. The Royal Road problem is also suitable for genetic algorithms because it makes use of heavy crossover applications. And it's very similar to the Max 1 problems. However, it involves n blocks of k bits versus strands. So it's going to be blocks of bits versus like a strand of like 0 and 1s. And so that makes it a little bit different. Another problem is the Cold War problem. So this deals with 2 populations that are simultaneously evolved. So it's basically, it's double of the Max 1 problem. We're going to run the same process in parallel with another population. Therefore, it evokes parallelism, as if an evaluation is done for pairs of candidates versus individual. So it's basically doubling what we did in Max 1s, and then also combining those separate populations that we get from the iterations. For this one, since we're dealing with parallelism, this is the part of like the advantage of using DNA implementation is that a computer implementation would have the time complexity squared, since we're doing 2 of these problems. But the DNA implementation allows it to remain unchanged because of its ability to compute in parallel form. So as a conclusion, based on everything we talked about, one of the most advantageous use of genetic algorithms is its ability to refine and discover high-quality solutions for presented problems. For example, the application I talked about, we were able to refine our candidates and amplify them and purify them to find a perfect one for the problem, and we could repeat the iteration for that problem to get the best answer possible. So obviously in the name genetic algorithms, it's based on and compared to existing studies based on genetics. This I see this as an advantage because then it kind of bridges the gap between, you know, the science of computer science and biology, where you can apply several concepts to existing ones in different fields. In addition, the application of these genetic algorithms can be used in various forms. We talked about a image improving one, which is a traditional computer-based problems and used a lot in things like machine learning. And we also talked about a problem that's directly relating to the use of actual DNA and recombining strands of DNA. And as discussed, we also talked about how genetic algorithms saves a lot of computing time for certain problems, especially a lot of problems that are described in like NP-hard categories. And that concludes the presentation. Thank you.